the lobes of the brain are named from the bones that cover them. In other words, here you have your frontal bone of your skull, so it's going to cover the frontal lobe. Parietal bone is the parietal lobe, the occipital bone, and so forth. All right, I want you'll look at, and obviously, different areas of the brain carry out different function. Here you see, we have sensory areas, we have motor areas, and if you look at this on page 291, you'll see some different areas, like in the temporal lobe, you'll see the auditory area. These are areas of the brain that are associated with us hearing. You'll see the visual cortex in the occipital lobe, back here. Okay, information comes in, light comes in through our eyes, stimulates photoreceptors in our eyes. Those tracks go to the back of the brain and we see. Same with smelling and all our senses. Different areas of the brain take on those functions. All right. All right. You've heard of that? What, men are right brain and women are left brain or something like that? All right, let's break the brain down then and just to its primary structures. All right, the brain. Largest part of the brain, called the cerebrum. All right, and you'll see there's all this. Then you have the cerebellum. And then the brain, or the diencephalon, or which incorporates the brain stem. Okay, and the brain stem consists of the midbrain, the pons, and the medulla oblongata. Each of those have particular function, but what I want you to sort of underline, maybe put an asterisk by, is the medulla oblongata, because there's areas in that brain stem that these are vital function areas. You'll see over here, you have the cardiac center, the vasomotor center, and the respiratory center. Okay, you can get a gunshot to the cerebrum. Okay, and if none of the brain stem is damaged, the body theoretically will stay alive. Cardiac center's working, the respiratory center's working. It's just that the light's on and nobody's home. So, you'll need to artificially feed and take care of all of those things that we would normally do consciously, but the brain stem, as long as it has not been damaged, will take care of, the basic body function. Okay? And there's different, the brain, the cerebrum is divided into lobes, and a important function of the cerebellum, which is this, it's often described as this cauliflower, bilobed cauliflower structure at the base of the brain, at the top of the brain stem, and it functions in coordinating voluntary action. Okay, and then like the frontal lobe, the prefrontal cortex of the cerebrum, this is where conscious thought takes place. This is where we generate action potentials to cause movement and things like that. A lot of our voluntary motions. There's other functions there. Unfortunately, because of time, we won't go through that. But I do want to say this, because this is important. Around the brain, well, let's say this. Inside the brain, we have these areas that are called ventricles. Okay? And what these, you have the lateral ventricles You have the third ventricle and the fourth. Now, why do I bring that up? Well, because in these ventricles is where cerebrospinal fluid, or CSF, spinal fluid is produced. Okay, and we're constantly producing this stuff and constantly reabsorbing it under normal conditions. But what that cerebrospinal fluid does is primarily it is a protective mechanism. It's mostly water. It comes from the water portion of our blood. Okay, it's formed in an area of these ventricles known as the choroid plexus. I'd write that term down. This is where CSF is formed. Okay, and then, I'm sorry?
It's formed in the...
In this specialized tissue that's, when we mentioned those ependymal cells earlier, they line the specialized tissue called the choroid plexus. And most of these choroid plexes are in the lateral ventricle, but there's a little bit in the third and a little bit in the fourth. And this is where cerebrospinal fluid is produced. Now that flows through these, it flows from the lateral to the third to the fourth, and then out in the subarachnoid space, which I'll mention in a minute, down the spinal cord, the spinal canal, and back up and around the brain. And it's a protective kind of thing. Okay, water is not very compressible. Okay, so when you get jolted in the head, that pressure being directed inward gets absorbed by that fluid, and we don't damage tissue. Okay, along with having bone there too. Bone being the primary, but then we have this watery protective mechanism covering the brain and spinal cord. Okay, now where was I going with that? The CSF, cerebrospinal fluid, is formed in the choroid plexes of the lateral third and fourth ventricles, and then it gets reabsorbed by a structure known as the arachnoid villi. And then I'll kind of expound on that in a minute, and we're constantly producing this stuff. It circulates, it's dynamic, it moves, it doesn't just stay still. We produce it in the choroid plexus, it gets reabsorbed in this arachnoid villi, and it circulates. Like if you have because you've had children, have you ever had a spinal anesthetic? Do you know? You've had an epidural maybe? Neither one of those. All right, well, if you have a spinal anesthetic, where they inject local anesthesia into your spinal canal. Well, where they're putting it is in this area known as the subarachnoid space where this cerebrospinal fluid circulates. And it's all volume controlled. If I put in so much volume, I'll get numb up to here. I put in a little more, I'll get numb up to here, and so forth, okay? What I used to do when I had to do, crash spinals for cesarean delivery, just to be nice, I'd put a little morphine in there so that the woman was comfortable. But you have to be very careful because this stuff circulates all up and down around the brain, and morphine is water soluble. And so if you do too much, then that morphine will circulate up around the brain and around the spinal nerves, around C2, where the nerve comes out that controls your breathing, and you knock out their breathing. You don't want to do that. But anyway, so this stuff is, for the most reabsorbed at roughly the same rate that it's produced. under normal circumstances. Now let me tell you what happened to my wife. She has what's called a acoustic neuroma. And this is, anytime you see that same OMA, you're thinking about a tumor. And this is on her acoustic nerve in her brain. And it was about the size of a tennis ball. And what it was doing was blocking the flow, the normal flow of this cerebrospinal fluid. So then what happens if you block the flow? If it can't flow out from the lateral to the third to the fourth, down, around, and back up around the brain, it starts to accumulate in one of these ventricles. In her case, it was the lateral ventricle. And so you start building up fluid in there and creates pressure. First symptom is going to be you're starting to get in a hell of a headache. Okay? So what they did for her was they put in a silastic tube, drilled a hole in her head, put a tube into her lateral ventricle, ran it underneath the skin and the skull, down her neck, down her side, and into her abdomen, into her peritoneal cavity. So that when the pressure started to build, there was a valve in that tube, and it would just go down that tube and into her peritoneal cavity and be reabsorbed. It's still there, still working.
And that worked for years?
Yeah, still working. And this happened 5 years ago. To keep that fluid from building up, because again, it's being constantly produced. Now, having said that, also around our brain and our spinal cord, we have a series of protective membranes known as the meninges.
Jeez.
I got to get to these, so I want to do these. All right, meninges. You know what you've heard of meningitis, right? That would be an inflammation thereof of these meninges. There are three. There's one called the pia mater, the arachnoid mater, and then the dura mater. Why do they call it that? Well, mater, sorry, I think the German word for mother is mater. All right? So we have these three meninges. That this pia mater is the innermost. It actually adheres to the surface of the brain and the spinal cord. Okay, so this is the innermost. Okay, and then between the the pia mater and the arachnoid mater is this space, and it's called the subarachnoid space. Okay, and this is where the cerebrospinal fluid circulates, in the subarachnoid space. Okay, then you have the arachnoid mater, because it's called that because what's an arachnoid? Do you know? Or what's an arachnid? A spider. Yeah. And so this membrane kind of looks like a spider web. Okay? And then above the arachnoid, okay, that's the middle. And the subarachnoid is below that. Okay, so here go from inner to outer, outer to inner. Right? This actually adheres to the surface of the brain and spinal cord. Underneath that is a space where the subarachnoid, where the cerebrospinal fluid circulates. Then we have this arachnoid mater, and then we have this dura mater. And I had asked Cassandra, did she ever have, when you had your children, did you have an epidural? Well, what an epidural is they take a needle, pretty big one, and they stick it in between your vertebrae, and they go to right about there before you hit the dura. And then they inject, and there is a a potential space there. That if you squirt something into it, the space will expand. And it will spread local anesthetic around the spinal cord. Okay? Now, one problem.
Do people with back pain get epidural shots?
Yes, I've had 3.
Oh, okay.
Because I've had, I ruptured 2 discs in my back, and then eventually 2 of the vertebrae broke. But before that happened, to help treat the pain with those ruptured discs, yes, I had 3 of those. And they stick that needle in right before the dura, and it's all feel. And then because you can feel it, you take this needle, it's a 16 gauge 2E needle, and put it on the end of a syringe, open up the syringe, and then you shove it in, and then you tap on the end of the syringe, and you advance it. It's all but blind. You can't see what you're doing, but you get a feel for it. And then at one point you go, the syringe will go in, you've hit that potential space, you take the syringe off, fill it up with local anesthetic, and squirt it in. Then you get numb, depending, and again, that's kind of volume control. But anyway, one of the problems with doing spinal anesthetics is that when you do a spinal, you're doing it different. You do it all the way into the subarachnoid space and squirt the local anesthetic. All right, now, a lot of women and men, anybody, say, I don't know if I want to have that epidural, because again, it's a blind procedure. It takes finesse. You have to learn how to do it. And if you take that big old needle and for some reason go a little bit too far and go into the subarachnoid space, spinal fluid can leak out and does leak out. And then you can imagine, because this adheres to the surface of the brain, and then these things kind of collapse, and you're talking about a hell of a headache. The so-called spinal headache. Okay, so how do they treat that? Well, they take that, because when you do a spinal, you use a very, very, very thin, 20 gauge needle. This one here for the epidural is a 16 gauge. The lower the number, the wider the bore. So anyway, how you treat it's not important, but that's the difference between those two. But what do we care about that? Remember the 3 names. These are the meninges. They go around the brain and the spinal canal. Now remember, we said the spinal cord in the average adult ends at about the level of the 2nd lumbar vertebrae. But the spinal canal goes all the way down to the sacrum, and so do these meninges. Okay, and so does the cerebrospinal fluid. All right? Oops. Okay, formed in the choroid plexus, and then at the top of the brain, between the dura and the arachnoid mater, I believe, if memory serves me correctly, there is that structure called the arachnoid villi, where that cerebral spinal fluid gets reabsorbed. Okay. You're going to just have to read about cerebrum, cerebellum. We have the 12 cranial nerves. Oh, before we quit, I want to bring this up. All right. We really didn't even have a chance to get a lot into the brain itself and the brain stem, so that you're just going to have to look over it. And the PowerPoint is really good on that because it focuses on the main components that we need to be concerned about. So do check out the PowerPoint, it's particularly on the brain. Now we have spinal nerves. Now look on page 301, and you're going to see those spinal nerves, okay? And they go from the first cervical vertebrae, C1, all the way down to S5, the 5th sacral vertebrae. And remember, we numbered the vertebrae when we talked about the bones. Start C1 at the top, and then all the way down to S5. Well, same with these spinal nerves. And there you can see they come out from the spinal cord. Your spinal nerves are broken into 2 divisions. There are what they call the sacral. All right, and these are the nerves that leave the spinal cord from the cranial nerves and the sacral nerves. Okay, and then you have the thoracolumbar division. All right, and these are the spinal nerves that leave through the thorax or the chest and the lumbar region. Okay. All right. What I had asked you to do in the, if you've looked at the discussion forum, is I wanted you to compare and contrast, or contrast mostly, the differences between the sympathetic and the parasympathetic nervous system. Okay? You'll see that the sympathetic nervous system, the spinal nerves that control the sympathetic nervous system are the thoracolumbar. Sympathetic, and the ones that control the parasympathetic are the craniosacral. Now, when you contrast them, all right, if you look at, when you read what I asked you to do, I want to know their relative size. So if you look in the sympathetic division, you have two types, in both of these divisions, you have two types of neurons, because they involve two neurons. You have a preganglionic, a ganglia is just a collection of nerve bodies, and you have a postganglionic. Okay, so these nerves, they come out of the spinal cord, those are the preganglion nerves, and then they will synapse with a ganglia, which is a collection of nerve cell bodies okay, they'll synapse with the ganglia, and then out of that ganglia you'll have postsynaptic. Out from these cell bodies you'll have another axon coming out, which is the postsynaptic neuron. And both the sympathetic and parasympathetic have both. Okay, and I guess what we need to focus on is the neurotransmitters that are used with each one. All right, preganglionic, sympathetic, SYM, and parasympathetic. They both use the same neurotransmitter, and it's acetylcholine.
They use what?
A neurotransmitter called acetylcholine. Actually, it's the same kind of neurotransmitter that's used at the neuromuscular junction, only it has a different effect here. So, both in the sympathetic and the parasympathetic divisions, oops, let me put this up here. The preganglionic neurons use acetylcholine. Okay. In the postganglionic neurons of the sympathetic division, the neurotransmitter is what they call norepinephrine, or noradrenaline, if you will. Okay.
Where's this at?
This is in the postganglionic neuron of...
No, I mean like for the discussion board, is it like we can go to the book to get all that?
Yes.
Okay.
The postganglionic neuron of the parasympathetic, it's easy to remember, I guess, because parasympathetic only uses one neurotransmitter, acetylcholine.
It's right there. That's the summary, and that's what I said. Right there.
Okay? Those neurons that use acetylcholine as the neurotransmitter are termed ACE. cholinergic. So, both the pre and post ganglionic neurons of the parasympathetic division, they're cholinergic because they secrete the neurotransmitter acetylcholine. Those neurons that use norepinephrine or noradrenaline are termed adrenergic. Because noradrenaline, norepinephrine is sort of a precursor to epinephrine, and the other name for epinephrine is adrenaline. So norepinephrine. So the only ones that, for our purposes, the only autonomic nerves that use or secrete epinephrine are the postganglionic neurons of the sympathetic division. And that makes sense, because what does adrenaline make you do?
Makes you.
Makes you hyped, get you up. Yeah, blood pressure goes up, heart rate goes up, everything. Okay? So, remember the pre and post ganglionic neurons. There's two in each division. One pre, one post. They have a different size. As an example, in the sympathetic division, the preganglionic neurons tend to be short because they come out of the spinal cord and they synapse with ganglia that are very near the spinal cord. So the preganglionic neurons are short, and then the postganglionic neurons tend to be longer. And then they go out and they synapse with different effectors. You know, and if you look, you can see the different effectors here: the eyes, the heart, and the lungs, and the liver, and the gallbladder, and all that stuff. In contrast to that, the preganglionic neurons of the parasympathetic division, and if you can see that here, they tend to be much longer. Okay, and then where they synapse, they either synapse at a ganglia right next to the effector or on the effector itself. So here, you can see.
So this is
This is sympathetic,
and this is
this is para. So you see the preganglionic neuron is short, preganglionic neuron is long. Postganglionic neuron is long, postganglionic neuron is short. So you want to address that. You want to address the neurotransmitters. And there's only two. I mean, acetylcholine and norepinephrine. Well, there's the other, but we won't get into it. And then some contrasting effects. A sympathetic response would be that your pupil will dilate. A parasympathetic response, the protective response, is your pupil will constrict so as not to damage your retina. A sympathetic response would be your heart rate goes up to pump more blood. A parasympathetic response would mean your heart rate comes back down.
Okay, I'm trying to find that over here.
Okay, you'll see these different responses.
There's a summary right here at the end that might have it.
Yes, I think there is
Right here, I'll show it's right.
Lee Heather, yeah.
And
What is this right here? Effects of neurotransmitter substance on visceral effectors. Here you have a sympathetic stimulation. Here you have a parasympathetic stimulation. For instance, in the pupil, sympathetic is dilation, parasympathetic is constriction.
So is that how you want me to do it? You want it?
Yeah, you don't have to go through all of them. Yeah, you don't have to. Just name a few.
Okay.
Okay? Bronchioles of the lungs, the breathing tubes. Sympathetic is they get bigger and let more air in. Parasympathetic is they get smaller
Okay.
because you don't need that much air. All right, and just kind of go through those. And what I was telling Tom earlier, I don't know if you were here, but think about what you want your body to be doing. If you're stressed and you're going to fight, you want your body to be doing stuff that's going to help you out. When you're not stressed, you want your body to be doing stuff that's going to protect you. You don't want to wear out your heart. You don't want to run a heart rate of 110, 120 for any great length of time. That sucker will give out on you. Okay, you don't want your blood pressure up there very high for any length of time. You don't want to blow a blood vessel. You want it to come back down, relax, calm, meditate, that kind of thing. Okay. I think they told me we gotta get out of here, but
They're not gonna kick us out because downstairs, they're trying to close the library, but up here,
we're okay?
we're on our honor system.
Okay, well, I got about 5 more minutes, so. All right, so
So we're doing chapters 9 and 10, right?
Yes.
That's what
nervous and
the quiz is
senses.
gonna be tomorrow?
Yes.
Okay. Nervous and sensitive.
Okay, now that was kind of the quick and dirty on the brain, or the brain and spinal cord. I guess for the senses, let's just kind of touch on the basics. You know, we see, we taste, we smell, right? And we feel. The special senses, we'll start with the tongue. Those types of cells that help us to taste are called gustatory. Taste buds, gustatory cells. We have 5, well, there was 4, but then they came up with this other one. 5 primary tastes, right? We have sweet, we have sour, we have salty, we have bitter, and then there's another one called umami, which is a Japanese word that means it tastes good, or it's kind of a meaty kind of flavor. All right, and we have these spread all over our tongue. Here's an important point to remember about taste. Anything that you taste, in order to be able to taste it, okay, the molecules of whatever it is you're eating and tasting and chewing up in your mouth, I mean, you want to be able to taste them, they have to be dissolved in saliva in order to taste it. Now, about our taste palate, because they're so close together, most of our taste palate is a combination of taste and smell. I don't know whether you've noticed it or not, but if you've ever noticed, if you have a cold or you're heavily congested, food just doesn't taste as good as it does if you're not. Because a lot of the taste palate that we develop over the years comes from a combination of taste and smell. Just like, okay, so for smell, those are olfactory cells. And just like with the gustatory cells, in order to smell something, that those molecules of whatever it is you're smelling have to be dissolved in mucus. And again, as I mentioned, most of our taste palate is a combination of that. You know, when you eat a Snickers bar, which happens to be my personal favorite, what you taste mostly is the sugar, right? The chocolate is more of an aroma, and that's what makes Snickers right on time. You know what I'm saying? But it's mostly you're tasting just the sugar that's in the candy itself, and then the chocolate is a lot of that is smell. That's why they're so good. Okay, so we have gustatory cells, we have olfactory cells. I don't know, is there much more I want to say about those without... I do want to just briefly look into the eye, and I want you to remember one thing about the eye, because I'm going to have to go